The word chemotherapy is defined as the therapeutic use of chemical agents to treat a disease. However, the common use of the word chemotherapy refers specifically to the use of chemical agents to treat cancer. Antimicrobial chemotherapy is the collective term for the therapeutic use of chemical agents to treat a disease caused by pathogenic microorganisms. Antimicrobial chemotherapy can be subdivided into antibacterial chemotherapy, antifungal chemotherapy, antiviral chemotherapy, or antiprotozoal chemotherapy, depending on the identity of the disease-causing microorganism to be eliminated. There are some key concepts that are fundamental to the understanding of how antimicrobial chemotherapeutic agents are developed and used. The mechanism of action refers to the specific biochemical interaction with target biomolecules through which an antimicrobial chemotherapeutic agent produces its microbicidal or microbiostatic effect. Target molecules can include enzymes or other proteins, nucleic acids, membrane lipids, cell wall, carbohydrates, and so on. The spectrum of activity refers to the taxonomic range of microbial types against which an antimicrobial chemotherapeutic agent is effective. Antimicrobial chemotherapeutic agents are typically classified as being either broad spectrum, meaning they are effective against a wide range of microorganisms, or narrow spectrum, meaning they are effective against a smaller set of microorganisms. The route of administration refers to the path by which an antimicrobial chemotherapeutic agent is taken into the body. The most common routes of administration for antimicrobial chemotherapeutic agents are oral, meaning the agent is swallowed, usually in the form of a pill, intravenous, meaning the agent is injected into the bloodstream, and topical, meaning applied to skin or mucosal membrane, usually in the form of a cream. In order to be effective, antimicrobial chemotherapeutic agents need to satisfy a number of criteria. They need to be potent, meaning they must show an effect at low concentrations so that an effective dose can be contained within 1 or 2 pills or a single injection. They need to be selective, meaning that the antimicrobial chemotherapeutic agent must specifically target a molecule or process in the pathogenic microorganism without affecting a similar or related molecule or process in the host organism. If a chemotherapeutic agent is not selective, this will result in toxic side effects in the host. An antimicrobial chemotherapeutic agent should preferably also target a wide spectrum of pathogenic microorganisms. An antimicrobial chemotherapeutic agent also needs to be safe in the sense that it does not cause an allergic response in the host. Nor should it produce harmful off-target side effects in the host that are unrelated to its antimicrobial mechanism of action. An antimicrobial chemotherapeutic agent also needs to be chemically and metabolically stable so that it can be stored for extended periods of time and also persist in the host organism after administration long enough to have the desired effect without being metabolized. The therapeutic index or therapeutic ratio is a useful way of quantifying the relative safety of an antimicrobial chemotherapeutic agent. The therapeutic index equals the dose at which at least 50% of the subjects display a toxic response, divided by the dose at which at least 50% of subjects display an effective response. Antimicrobial chemotherapeutic agents differ widely in their therapeutic indices. For example, antibiotics belonging to the beta-lactam and macrolide families have wide therapeutic indices. Conversely, the antibiotic vancomycin is a good example of an antimicrobial chemotherapeutic agent with a narrow therapeutic index. Antimicrobial chemotherapeutic agents with narrow therapeutic indices are therefore only used when safer chemotherapeutic agents are not effective or otherwise not available. In such instances, patients must be closely observed for potential toxic side effects. Chemical compounds that have antimicrobial activity can be derived from a number of sources. By far the most common source of antimicrobial chemotherapeutic agents is microbial secondary metabolites, which include some of the most widely used antimicrobial chemotherapeutic agents today. Plant secondary metabolism is a source of some prominent antimicrobial chemotherapeutic agents, especially antimalarial agents. Animals produce antimicrobial peptides either as body secretions in invertebrates, but also by immune cells of all animals. Although animal antimicrobial peptides appear promising, they are not in widespread clinical use so far. Antimicrobial chemotherapeutic agents can also be produced through chemical synthesis. One such approach is to chemically modify naturally occurring antimicrobial compounds to, for example, improve their potency or decrease their toxicity. Chemical synthesis can also be used to produce entirely novel structural classes of compounds with antimicrobial activity. The remainder of this presentation is divided into 5 sections. The first 4 sections will explore chemotherapeutic strategies against pathogenic bacteria, fungi, viruses, and protozoa, and give representative examples of individual chemotherapeutic agents for each group of pathogens. The final section will discuss mechanisms by which microorganisms can become resistant to antimicrobial chemotherapeutic agents. This section will also discuss some of the larger issues regarding antimicrobial resistance, as well as some of the methods used to determine susceptibility to antimicrobial chemotherapeutic agents. Bacteria are omnipresent in both natural and man-made environments. Bacteria also colonize the human body, both as beneficial symbionts as well as neutral commensals. Because of the evolutionary distance between bacteria and eukaryotes, there are many potential chemotherapeutic targets within the bacterial cell. The short generation time of bacteria and their tendency to exchange resistance genes through horizontal gene transfer poses a great challenge in the development of new antibiotics. The double membrane of gram-negative bacteria is an additional challenge, as this is an effective barrier against many antibiotics. Chemotherapeutic targets in bacteria include the bacterial cell wall, which has no counterpart in eukaryotic cells. The core cellular machinery, which includes such processes as DNA replication, mRNA synthesis, and protein synthesis. The evolutionary distance between bacteria and eukaryotes make these processes good targets for antibiotics. However, it should be noted that the mitochondrial ribosome is of bacterial origin and can therefore be more sensitive to antibiotics than the cytosolic ribosome. There are also essential metabolic pathways in bacteria that do not have counterparts in human cells. One such pathway is the folic acid biosynthetic pathway in bacteria, which is the target of sulfonamide antibiotics. Here is an overview of the spectrum of activity for a selection of antibiotic families. Note that some antibiotics, such as tetracyclines, have a broad spectrum of activity, while others, such as vancomycin, have a quite narrow spectrum of activity. Also note that some bacterial groups, for example the mycobacteria, are only susceptible to a limited number of antibiotics. Now we will look at some representative examples of antibiotics from different structural families. Penicillin G is a beta-lactam family antibiotic that targets bacterial wall synthesis by inhibiting the formation of peptide crosslinks between glycan chains. Vancomycin is a glycopeptide antibiotic that also inhibits the formation of peptide crosslinks between glycan chains in the bacterial cell wall. However, due to its large size, it cannot penetrate the outer membrane of gram-negative bacteria and is therefore only effective against some gram-positive bacteria. Isoniazid is an inhibitor of cell wall biosynthesis in mycobacteria by specifically blocking the synthesis of the cell wall component mycolic acid. Daptomycin is a lipopeptide antibiotic that integrates into the bacterial membrane, which leads to the formation of pores. These pores then lead to the leakage of small ions in and out of the cell, which disrupts the membrane potential. Chloramphenicol belongs to the Amphotericin family of antibiotics and acts by inhibiting bacterial protein synthesis. Chloramphenicol also affects mitochondrial translation in human cells, which is why this antibiotic is typically only used externally. Erythromycin is a macrolide antibiotic that also inhibits bacterial protein synthesis. Doxycycline is a tetracycline antibiotic with a broad spectrum of activity. Tetracyclines also inhibit bacterial protein synthesis. Rifampicin is an example of an antibiotic that targets bacterial mRNA synthesis by binding to the bacterial RNA polymerase. Sulfamethoxazole is a sulfonamide or sulfur antibiotic which blocks the synthesis of folic acid by inhibiting one of the enzymes in this pathway. One of the consequences of this inhibition is that the synthesis of the DNA-based thymidine is inhibited, which prevents the bacterium from replicating its genome. Since humans get their folic acid through diet, sulfonamide antibiotics do not affect this process in human cells. Trimethoprim is another inhibitor of the bacterial folic acid biosynthetic pathway, but targets another enzyme in the pathway. Sulfamethoxazole and trimethoprim are typically used together to decrease the chance that a bacterium will develop resistance to one of the antibiotics. As with bacteria, fungi are also omnipresent in both natural and man-made environments. Fungi, mainly yeasts, can colonize the human body, but do so as commensals rather than symbionts. Fungal infections are typically opportunistic in nature and occur when the human immune system is weakened due to stress or disease. The main challenge of antifungal chemotherapy is the close evolutionary relationship between animals and fungi, which limits the range of chemotherapeutic targets in fungal cells. Chemotherapeutic targets in fungi include the fungal cell wall, which has no counterpart in mammalian cells. The steroid biosynthetic pathway, which is subtly different in fungi compared to animals. The main steroid in the fungal plasma membrane is ergosterol rather than cholesterol. This figure shows the subtle structural differences between cholesterol and ergosterol. Structural differences are highlighted in red. Fluconazole is an example of an antifungal agent belonging to the azole family. All azole family antifungal agents target the enzyme 14-alpha-demethylase, which is involved in steroid biosynthesis. Although this enzyme also exists in human cells, it is sufficiently divergent to not be significantly affected by the antifungal compound. Amphotericin B targets fungal cells by directly binding the ergosterol molecule, which leads to leakage of ions in and out of the fungal cell. This compound also binds somewhat weaker to cholesterol, but this binding is strong enough to cause toxic side effects. Echinocandins are cyclic peptides that block fungal cell wall synthesis by inhibiting the enzyme beta-glucan synthase. The peptide backbone in the three echinocandins shown here have been highlighted in red to better show the cyclic structure. Viruses differ from other microbial pathogens in that they are not cellular organisms, and the basal machinery for genome replication and gene expression is typically very different from human cells. This makes them good targets for antiviral chemotherapy. The main challenge of antiviral chemotherapy is the accelerated mutation rate in viral genomes, which drives the evolution of resistance. Retroviruses such as HIV and herpes simplex virus pose an additional challenge by being able to integrate into the host genome. As long as the viral infection is latent and no active viral replication is occurring, the virus is resistant to any antiviral agents that target the viral replication cycle. Chemotherapeutic targets in viruses include the viral genome replication machinery. The divergent nature of most viral genomes mean that the virus requires its own enzymes to replicate the genome. These enzymes are good targets for nucleoside analogues. Many viruses express their genes as a single RNA transcript that is then translated into a single polypeptide, which is then cleaved into individual functional proteins by a viral protease. Such viral proteases are therefore good targets for antiviral chemotherapy. Isoniazid is an example of a nucleoside analog which is used to treat HIV infections. Glecaprevir is an example of a protease inhibitor which is used to treat hepatitis C infections. Oseltamivir is neither a nucleoside analog nor a protease inhibitor. Instead, it inhibits the influenza neuraminidase enzyme, which prevents the influenza virus particle from exiting the human cell after replication. Protozoa, also known as protists, are a very diverse group of predominantly microscopic eukaryotes. Because of this diversity, there are as of yet no truly broad-spectrum antiprotozoal chemotherapeutic agents. However, some antibiotics and antifungals also have antiprotozoal activity. A relatively small number of protozoan species are known to infect humans. These infections are often chronic in nature, such as malaria. Parasitic protozoa often have complex life cycles that can complicate antiprotozoal chemotherapy. Shown here are 10 of the most common protozoan parasites that infect humans. Note the diversity in morphology as well as in taxonomic placement among eukaryotic lineages. Listed here are some of the same protozoan parasites along with the diseases they cause, as well as some of the standard chemotherapeutic agents that are used to treat these diseases. Malaria is caused by the parasite Plasmodium and kills hundreds of thousands of people every year. Quinine was the first effective antimalarial chemotherapeutic agent ever discovered. The Plasmodium parasite specifically infects red blood cells and uses the hemoglobin inside the red blood cells as nourishment. During the digestion of hemoglobin, toxic heme is released that must be neutralized by the Plasmodium parasite. Quinine acts by blocking this detoxification process and thereby kills any Plasmodium parasites that are susceptible to this compound. Chloroquine is a synthetic antimalarial agent that also inhibits the detoxification of heme by Plasmodium. Artemisinin is also an antimalarial chemotherapeutic agent derived from the herb sweet wormwood. Its exact mechanism of action is not fully understood. Metronidazole is a synthetic antiprotozoal compound that acts by being converted into a toxic nitrosyl radical once it is inside the cell of the parasite. Paramomycin is an aminoglycoside antibiotic that also has antiprotozoal activity. It has been shown to inhibit protein synthesis by the cytosolic ribosomes in some protozoan parasites. The final section will discuss the development of resistance to antimicrobial chemotherapeutic agents by microorganisms. The main challenge for the effective lifespan of all antimicrobial chemotherapeutic agents is the short generation time of microorganisms, which ranges from days to minutes. If we can better understand how resistance develops, this will help both to effectively use existing antimicrobial chemotherapeutic agents for as long as possible, as well as developing new ones. There are two categories of resistance to antimicrobial chemotherapeutic agents. Intrinsic resistance means that a microorganism possesses an innate ability to resist the antimicrobial chemotherapeutic agent. This can be the result of structural or functional characteristics of the microorganism. The double membrane structure of gram-negative bacteria is one mechanism of intrinsic resistance to a number of antibiotics. Acquired resistance is an ability that is gained through an evolutionary event where a previously susceptible microorganism becomes resistant. There are a number of processes whereby microorganisms can acquire resistance to antimicrobial chemotherapeutic agents. The simplest is through spontaneous mutation. Resistance can also be acquired through exchange of genetic material through hybridization or horizontal gene transfer events. We will now briefly consider some possible cellular resistance mechanisms to antimicrobial chemotherapeutic agents. The simplest way to think of an antimicrobial chemotherapeutic agent is that it will interact with a component of a microbial cell. In this particular diagram, the compound enters the microbial cell and interacts with its target molecule, perhaps an enzyme or other protein. This interaction somehow disrupts the function of the target molecule. In the case of an enzyme, it can prevent a chemical reaction from progressing. The disruption of the cellular process leads to some form of cellular damage. The cellular damage can in turn lead to the death of the cell. One simple resistance mechanism is to simply alter the target molecule through some form of spontaneous mutation. Another possibility is that the microbial cell simply produces more of the target molecule so that there is still some residual activity in spite of the antimicrobial compound. The microbial cell may also bypass the blocked process step by, for example, using a similar target molecule that is insensitive to the antimicrobial compound. If the antimicrobial chemotherapeutic agent works by inhibiting a metabolic pathway, the microbial cell can also bypass this inhibition by acquiring a downstream metabolic intermediate from the external environment. The microbial cell can also counteract the cellular damage done by the antimicrobial chemotherapeutic agent by enhancing cellular repair mechanisms. Another mechanism to resist an antimicrobial chemotherapeutic agent is to simply inactivate it either by modification or digestion. The antimicrobial chemotherapeutic agent can also be sequestered to a cellular compartment where it is unable to exert its antimicrobial activity. Some antimicrobial chemotherapeutic agents require activation by the microbial cell. If this activation step is somehow lost or weakened, there will be little or no active compound to exert its antimicrobial activity. Antimicrobial chemotherapeutic agents that need to act inside the cell can be partially or completely excluded if the entry point is modified or lost. Lastly, the microbial cell can simply expel the antimicrobial compound by active transport out of the cell. The challenge of antimicrobial resistance derives from the long evolutionary history of microorganisms using antimicrobial compounds against each other in competition for limited resources. This has led to a very rich genetic reservoir of antimicrobial resistance genes in the natural environment, which is known as the resistome. The antibiotic resistome is of particular concern since bacteria are able to effectively and rapidly exchange genetic material across species barriers using horizontal gene transfer. This is reflected by the quick appearance of antibiotic resistance for any new antibiotic shortly after it enters clinical use. We will end by looking at methods for assessing antimicrobial resistance in microorganisms. The minimum inhibitory concentration is a quantitative measure of antimicrobial resistance which is typically determined using a broth dilution test. The microorganism is inoculated into parallel samples of liquid growth medium with a gradually decreasing concentration of the antimicrobial chemotherapeutic agent. The minimum inhibitory concentration is defined as the lowest concentration of antimicrobial compound where no growth can be detected. The Kirby-Bauer disc diffusion test is a qualitative test for antibiotic susceptibility where bacterial isolates are scored as either susceptible, intermediate, or resistant. An agar plate is seeded with bacteria and paper discs impregnated with antibiotic are placed on the agar surface. Antibiotics inside the discs will diffuse out into the agar medium, which leads to visible zones of inhibition if the bacterial isolate is susceptible to the antibiotic. The Kirby-Bauer test is scored by comparing the diameter of the zone of inhibition to a reference document. The Kirby-Bauer test can also be used to score antimicrobial resistance in pathogenic yeasts. A variant of the Kirby-Bauer test is to use antimicrobial gradient strips. These strips are applied to an inoculated agar surface as before. The minimum inhibitory concentration can then be determined by where on the gradient strip the zone of inhibition terminates as indicated in red.